Hello, my name is Marc de Meyer and I'm an entomologist specialized in the identification, taxonomy and systematics of African flies, in particular Tephritidae fruit flies. I work at the Royal Museum for Central Africa, which is based in Teveur, Belgium. This presentation deals with an introduction to the main pest groups of fruit flies found worldwide. It is part of a series developed within the framework of the FFF Fruit Fly Free project, supported by the Standards and Trade Development Facility. In this presentation, I will provide some general background on each of the main genera, how to differentiate them from each other using morphological characters of the wing and general appearance, and the common features for members of each group that can be used to recognize species within the genus. For characters on how to recognize the family as a group, I refer to the separate presentation with a general introduction. As indicated in this general introduction, the family Tephritidae or true fruit flies is divided into genera. Worldwide, there are about 500 genera. In Africa, about 160. However, only 6 of these are of economic significance. Of these, 4 occur in Africa. You see the 6 genera listed here. The genus Anastropha is a group found in tropical America. Species are found mainly throughout the whole of South and Central America up to Mexico. No species are reported as invasive outside the Americas. The genus Regulatus is a group mainly present in temperate climates of North America, Europe and throughout the Palearctic. A few species are also found in more southern parts of the Americas. Some species have been accidentally introduced into other parts of the world, in particular in North America and Europe. So far, there have been no introductions in other geographical areas. The genus Tephritidae is an African group, restricted to the Afrotropical region, meaning the area of Africa below the Sahara Desert. Some species are also found on the islands in the Western Indian Ocean, as well as those island groups in the Atlantic Ocean close to the African mainland. One species is an important invasive one, the Mediterranean fruit fly Tephritidae capitata, which has been introduced in several other parts of the world. The genus Dacus is also a predominantly African group, but several species are also indigenous to the Asia-Pacific region. Very few are invasive. Some have probably been introduced from the African mainland into some of the islands in the Indian Ocean, and one species, Dacus ciliatus, has spread from Africa into Central Asia. The genus Bactrocera is the most species group of all and is mainly found in the Asia-Pacific region. A few species are also indigenous to Africa. Several species in this group are considered invasive, the most notorious one being Bactrocera dorsalis. Currently, some Bactrocera species have been introduced into Latin America, the Mediterranean region, and islands of the Pacific Ocean. Finally, the genus Zogodacus is also a mainly Asia-Pacific group. Until recently, it was considered as a subgroup of the genus Bactrocera, but is now recognized by most researchers as a separate genus on its own. One species is invasive, Zogodacus cucurbitae, and has spread from Asia into Africa and islands in the Pacific Ocean. These 6 genera can firstly be differentiated into 4 groups based on the shape of some cells in the wing. The terminology and position of veins, cross veins and cells in the wing pattern in fruit flies is explained in the presentation on morphology, and I'll refer you to the latter for further details. The differences in the main groups are mainly with regard to the basal cubital cell and the basal medial cell. Their position is shown encircled in red in the image shown here. The basal cubital cell is abbreviated by BCU. Anterior of this is the basal medial cell, abbreviated by BM. The cell BCU has at the end a kind of finger-like appendage, while the cell BM is without such extension, but truncated at the end. The appendage is then prolonged as a vein, and the combined analcubital vein A1 plus CU2. The differences between the groups is mainly related to the specific shape of this extension, as well as to the size of cell BM compared to cell BCU. These differences will be explained in further detail in this presentation. Both cells are demarcated by a red circle or oval in the four main groups here. Clockwise from top left, you see a typical wing of a Tephritidae species. Next to it, you see the wing of a representative of a Dasein. This is the group which comprises the genera Bactrocera, Dacus and Zogodacus. All three genera have a similar wing morphology. Below this, at the bottom right, you see the typical wing of a Rhagoletis species, and next to it, below left, the wing as usually seen in Anastropha species. Now I will present each group a bit more in detail and show the difference in shape of cells BCU and BM. First, we present the genus Anastropha. As indicated, this group is mainly found throughout the whole of South and Central America up to Mexico. Several species are also found on the islands in the Caribbean. A few are present in some southern states of the USA. No species are reported as invasive outside the Americas, but some have expanded their range within the Americas. They are also frequently intercepted in fruits being imported to other parts of the world. More than 200 species are described within the genus, of which about 15 are considered of economic significance. Some of the species are considered to be a complex of different cryptic species, the most studied one being the Anastropha faturculus species complex. And some are polyphagous, attacking a wide variety of different hosts. Others have a more restricted host range, such as Anastropha grandis, which attacks cucurbids. Previously, the papaya fruit fly was placed in a separate genus called Toxotripana. It was called Toxotripana curvicauda. Recent studies showed that this and related species belong to the genus Anastropha. Most Anastropha species have a similar type wing marking consisting of bands. From the base to the apex of the wing, there is a sinusoid band running which in the latter part runs along the anterior margin of the wing. This is called the S-band. Below that one, there is a forked band which looks like an inverted letter V and is called the V-band. The wing markings are usually yellow-brown as shown here, but can sometimes be more blackish. Most Anastropha species are relatively large with a yellowish to brown-black body color, often marked with longitudinal stripes on the thorax. The female of some species have a very elongated ovipositor. Regarding the shape of the cell BCU and BM, the green double-headed arrows show the width of cell BCU versus BM. You can see that both cells have approximately the same width. The length of the finger-like appendage of cell BCU, this is from its base to the main body of cell BCU till its apex, is indicated by a double-headed red arrow. You notice that this length is approximately the same as the distance from the tip of the appendage till the wing margin. The dashed red arrow shows the length of the base of cell BCU, which is also more or less equal. We will compare these three dimensions with that in the other group. This slide shows a representative of the genus Tephritidae. Representatives of this genus are solely found in sub-Saharan Africa and on the adjacent islands. One species, Tephritidae capitata, has been introduced to other parts of the world, with the first introductions dating back more than 150 years. First in the Mediterranean region, but later also in other continents. About 100 species are described in this genus. Of these, about 10 are considered of economic significance. As with Anastropha, some species are polyphagous, while others have a more restricted host range, often confined to species of the same plant genus. Tephritidae species are usually rather small in size, with a similar wing marking consisting of a number of streaks and spots in the basal part close to the body and a number of yellow-brown to black-brown bands on the wing. The thorax and abdominal color may vary from pale yellow to black, with or without conspicuous spots. Regarding the shape of cell BCU and BM, also here the green double-headed arrows indicate that the width of cell BCU versus BM is approximately the same. The length of the finger-like appendage of cell BCU is distinctly shorter than the distance from the tip of the appendage till the wing margin, shown by the red arrows. It has a typical shape with a constricted or narrowed median part. The appendage itself is about the same length as the base of the cell, the latter indicated by the dashed red line. Representatives of the genus Rhagoletis are small flies, usually with distinct black wing markings and black to brown coloration on thorax and abdomen. There are about 70 species known worldwide. Although their occurrence seems to be in temperate regions, mainly of the northern hemisphere, but also some in the southern hemisphere of the Americas. As mentioned, there have been some species introduced on both parts of the Atlantic Ocean, thus between North America and Western Europe. These introductions are mainly on species attacking cherries and walnuts. Rhagoletis species are usually stenophagous, meaning that they attack a limited number of host plants that are closely related to each other. Some of them attack specific crops of economic significance and are considered pest species. It concerns here mainly apples, cherries, and some other soft fruits such as blueberries. The emergence of the flies therefore need to be synchronized with their host so that the females are gravid when it's the right time for fruit availability. Most Rhagoletis species are rather small and have a black or brownish body color, and with a contrastingly colored scutellum. They have a typical marking on the wing consisting of a number of transverse bands which are usually black colored. Regarding the shape of the cells BCU and BM, the green double-headed arrows show the width of both cells. Also here, both have approximately the same width. However, the appendage of cell BCU is very short and tapering towards a pointed apex, not like in Tephritidae where we saw it was constricted in the middle. The length of the appendage is distinctly shorter than the distance from the tip of the appendage till the wing margin. Look at the red arrows. And the appendage itself is also shorter than the base of cell BCU. Finally, there is the group of the daisines. This is a joint name for the representatives of the genera Bactrocera, Dacus and Zogodacus. In total, they comprise close to 1,000 different species, occurring in different parts of Africa, Asia and Oceania, including Australia and several islands of the Pacific Ocean. The table below left show you the numbers for each genus and their relative diversity in Africa versus the Asia Pacific. Here you can see that the genera Bactrocera and Zogodacus are predominantly Asia Pacific, while the genus Dacus is predominantly African. Several of them are known as invasive ones, the most notorious one being the oriental fruit fly Bactrocera dorsalis, the carambola fruit fly Bactrocera carambolae, the solanum fruit fly Bactrocera latifrons, the cucurbit fruit fly Zogodacus cucurbiti, the olive fruit fly Bactrocera olei, and the pumpkin fruit fly Dacus ciliatus. We will present them more in detail in a separate presentation on species of economic significance. All daisines have a similar morphology. They vary in size, but all have a rather elongated body, which can be black to pale brown in color. The wings are somewhat elongated. A typical daisine has some distinctive dark brown wing markings along the anterior margin of the wing, ending in a broader spot at the wingtip. At the base there can be a diagonal darker stripe present or absent. In addition, there can be other spots or marks on the wing as well. Daisine fruit flies have different feeding strategies, with being specialized on a single host, like the olive fruit fly, to being very polyphagous. Regarding the shape of the cell BCU and BM, the green double-headed arrow indicate clearly that the cell BM is much wider than the cell BCU, about twice as wide. The appendage of cell BCU is very long and narrow and distinctly longer than the distance from the tip of the appendage till the wing margin, the red arrows. Both the appendage and the base are about the same length. To differentiate between the three daisine genera, Bactrocera, Dacus and Zogodacus, one has to look at other morphological characters. Two characters allow you to split up these three genera into two groups, Dacus on one hand and Bactrocera and Zogodacus on the other. The position of these characters are shown in this slide. One deals with the absence or presence of prescutellar bristles. The position of these bristles is indicated with a red circle on the left-hand image. If present, they are placed anteriorly of the scutellum. The second character deals with the tergites of the abdomen, whether they are fused together or separate. The state of this character is observed the easiest along the lateral margin of the abdomen, the region encircled on the right-hand image. Here we see the two alternative character states regarding the prescutellar bristles. On the left-hand side, one sees the areas in dorsal view on top and lateral view below, when the prescutellar bristles are absent. The area where one would expect them are encircled in red. The prescutellar bristles are absent in all Dacus species. The left-hand images are therefore typical of the character state in Dacus. On the right-hand side, we see the same area, but then in representatives of Bactrocera and These are present as a pair of long black bristles. This state is typical of all Bactrocera and Zogadacus species found in Africa except for one, Bactrocera olei, the olive fruit fly. This is the only Bactrocera species found in Africa that is also missing the prescutellar bristles. The following slide deals with the second differentiating morphological character, the abdominal tergites. On the left-hand side, you see the abdomen in lateral view. The image on the right-hand side shows a close-up of the lateral margins. The top images are typical of all Dacus species. Here you see that the different abdominal tergites are fused together, forming one smooth carapax. The margins of the different tergites are fused to one another and the margin forms one smooth line. This state is found in all Dacus species. The bottom row shows the state in Bactrocera and Zogadacus species. Here the abdominal tergites can still be seen as separate entities. The margin does not form a single smooth line, but is made up of different components that are slightly overlapping each other. This state is present in all Bactrocera and Zogadacus species, including Bactrocera olei. So there are no exceptions found among the representatives of these genera in Africa. Finally, there's a distinction between Bactrocera and Zogadacus. Until recently, Zogadacus was considered a subgroup or subgenus of Bactrocera. Recent studies have provided evidence that Zogadacus should be considered as a separate entity and recognized as a separate genus. The genus Zogadacus has only one representative in Africa, the introduced Zogadacus cucurbitae, previously referred to as Bactrocera cucurbitae. This species can be differentiated from all true Bactrocera species found in Africa by the coloration of the thorax. The top image in the middle shows the general habitus of Zogadacus cucurbitae in dorsal view, and the image on the left shows a close-up of the thorax. In the posterior half of the thorax, there are three longitudinal yellow lines, two along the lateral sides and one in the middle. The presence of this medial line is typical for Zogadacus cucurbitae. On the bottom image, in the middle, one sees a typical Bactrocera species, while the image on the right-hand side shows a close-up of the thorax. One sees that the medial stripe is missing. This is typical of all Bactrocera species found in Africa. The lateral yellow stripes can be present as in the image here or also absent. The medial stripe is present in some Dacus species, but then the other character earlier mentioned regarding the prescutellar bristles and the fusion of the abdominal tergites will allow you to differentiate those from Zogadacus cucurbitae. Many thanks for your attention. This presentation was created in collaboration with Stellenbosch University. We would like to thank the funding agencies as well as the individuals and institutions who in one way or another contributed to the actual development of this presentation.